Okay, so sorry, I just banged my knee. So before we get started on the slides, we're going to go through some questions that were on the discussion forum. So we had some questions about the types of post-translational modifications and regulation. So someone asked what the different types of regulation there are, the three types, and this shows the three types. So allostery, post-translational modification, and nucleotide binding and hydrolysis. So we're going to quickly go through these. And then nucleotide binding and hydrolysis, we had a few questions on, so we're going to explain that a little better. So allostery, Iris, you want to explain allostery?
Yeah, so allostery is basically when you have a molecule that it binds to the protein, but it binds somewhere other than its active site. And it's basically changing the conformation of the protein, and it can change the conformation of the protein either in a way that can open the active site and allow it to become active, or inhibit the active site and therefore like prevent it from being activated, basically. So I think this diagram is really helpful. Like you can see under activation, like part A, the green thing is the molecule that's binding to the protein. And when it binds, look at like the next picture over, the protein changes conformation, it changes shape. It allows another molecule to go and bind to the active site. And now that the molecule is bound, the protein can be active, perform its function. And then
you just want to touch on, you said conformational state. We had a question about that. Do you just want to explain what you mean by that?
Yeah, so conformational state is basically the shape of the protein. So when a protein changes conformational state, it means like something in its chemistry has changed, possibly like now like a new R group is exposed, and that will allow another molecule to bind to that exposed like R group. Is that a good explanation?
Yeah, so in all of these pictures, the second picture is a conformational change. So like the protein started by looking like this, and then we put something in, and now it looks like this. So that's I have a question. Is there a specific name for that green molecule? No, these are yes, wait, what's the green molecule that comes in? It's a oh my gosh, they're not competitors. No, they're just proteins that come in. I mean, they can have different names. Sometimes they're competitors. So what's it called when, oh my gosh, this is 151. You did 151, right? So what is it when it's a non-competitive inhibitor, right? So when it binds somewhere other than, for
Possibly.
your intents and knowledge, so this is a non-competitive inhibitor, but for your intents and, what am I saying? This is great. This is awesome. We both just forgot English at the same time. No, in this case, you don't need to know. You just need to know it's another protein that comes in to a different site other than the activation site, changes the conformation, and either activates or inhibits the protein activity.
Yeah, so conformation is just the shape of the protein.
So when it says change conformation, it changed shape.
Yeah.
And then some questions on post-translational modifications, examples. So if you want to go through these three examples, Iris.
Yeah.
Just maybe talk about ubiquitination, phosphorylation, just what those are.
Yeah, so with ubiquitination, this can happen, or this happens after the protein has already, reached its final stage of folding. In the case of ubiquitination, sometimes it means that the protein will get sent to the proteasome, right?
Mm-hmm.
Yeah. Okay.
So we stick a ubiquitin.
Ubiquitin.
Yeah.
And so that signals to other molecules that the protein should be degraded. In the case of phosphorylation, it's the same idea where it's like after the protein's already reached its final stage of folding, if it's phosphorylated, it can allow other proteins to bind to it. And
Can
I'm trying
activate
to
or
think.
deactivate.
Oh,
Phosphorylation is
yes.
one of the very general ones that can do a lot of things. It's when we just stick a phosphate group on it, and you'll see that a lot in this class.
Yes, it will come up a lot, and it can either be an activating phosphate or an inhibitory phosphate. Acetylation, I'm sort of
You know,
blanking on.
we don't really talk, we just stick an acetyl on it, but we don't really talk about acetylation. I wouldn't worry too much about it. Just know that you can kind of stick all these groups on. So you
Yeah,
can stick an acetyl on it and it changes, it sends some sort of signal or changes some sort of binding so that the protein changes,
yeah,
but they're already, in all of these cases, they're fully folded already. And you can stick this thing on or take it off. It's not going to change the actual way the protein's folded, but it can affect how the protein is functioning.
yeah. It changes like the chemistry around the protein, and so it can change the shape of the protein, which can also change its function.
Yeah.
And like
then
how
the
it
last
interacts.
one, this is the one we had the most questions on. So if we want to walk through nucleotide binding and hydrolysis in the case of GTP and GDP.
Yes. Okay. All right. Let me see. So I'll start with the diagram with the GDP.
This one
Yeah.
in here.
All right. So when GTP is bound to this protein, the GTP binding protein, it's considered active. The third phosphate in GTP can be cleaved, which changes it to GDP. So GTP is guanosine triphosphate. It has 3 phosphates. GDP is guanosine diphosphate, and it has 2 phosphates.
And the guanosine is the
when
nucleotide here. I think that might also be confusing people, that the G is the nucleotide, you know, like ATCG. So that's the nucleotide binding and hydrolysis that's happening.
Right. When it's in its GDP bound state, it's inactive. I don't really know what this next step is showing.
So GDP dissociates either very slowly or most of the time we're going to talk about requires assistance, or it'll dissociate very slowly by itself. But in the pathways we talk about, we need something to help remove it, an enzyme. And GTP has very high affinity for this protein, so that when GDP dissociates, GTP comes in very quickly and turns this on until something comes, a phosphatase is the enzyme is called, and it cuts off the phosphate. That's the hydrolysis. So nucleotide binding is the GTP, and the hydrolysis is when we go from GTP to GDP. And ATP is the same thing. And so this is showing you that this enzyme, ATP, comes in with 3 phosphates. Somehow there's a conformational shift. We cleave the phosphate, and then we end up with ADP, only 2 phosphates. So that's what it's showing you, that this is a RAS. No, this is ATP. So this is some sort of enzyme. It's a phosphatase, so it removes a phosphate. So when we're talking about nucleotide binding or nucleotide type switching, this is what's happening, is that your GTP or ATP is switching with ATP or ADP or GDP.
Yeah, and remember that every time GTP changes to GDP or vice versa, it changes the conformation of the protein. Whenever the protein conformation is changed, its function is changed too.
Yeah, which is why it's on with the GTP and off with the GDP.
Yeah.
Sorry, I thought you were done with your sentence. I didn't mean to cut you off.
No, that's totally fine. That was a good addition.
But yeah, so the GTP bound state the conformation is changed. And so for some reason that turns on the protein. And that's why it turns it off when it's GDP, because we're missing that phosphate like here, and suddenly it's a different shape. So that's, and then someone just had a question about an enzymatic step. The enzymatic step is just steps that enzymes are involved in to make something go faster. So if like GDP, we send in an enzyme to help it get off quicker, that would be an enzymatic step. It's a step where an enzyme is involved. And then the last questions we have are a transient interaction. Someone wanted me to define a transient interaction. This has to do with like non-covalent bonds. Transient just means not permanent. So covalent bonds are essentially, for all intents and purposes here, kind of permanent bonds. It's very hard to break them, versus transient means they last a little while, but then they break. So transient is just when, I don't know if you've ever heard the word of a transient person. It's just not there forever. Ionic versus ion dipole is something people are struggling with as well. An ionic bond forms between two ions. So this means that one is charged positively and one is charged negatively. Those are ions. Ions have a permanent charge on them. They're missing an electron or two, or have an extra electron or two. Versus an ion dipole, if we remember correctly, a dipole movement is when something is partially negative or partially positive because it's a polar molecule. So like OH is polar, the O is slightly negative and the H is slightly positive. So that slightly negative O can make an interaction with a charged positive. It's not an ionic bond, but they can interact because there is a difference in charge there, and they are attracted, just not as attracted as two full ions. So that would be an ion dipole, because one side is an ion and one side is a dipole. So that's the difference between those. And then the last question we had, oh, by the way, some of these were problem set questions in the forum. I will tell you that I personally do not answer problem set questions until I stop recording. And that is to encourage people to come to the review sessions. So if you come and you stay, I will finish the recording and then we can do problem set questions. But I don't want to just give problem set question answers or help to people that aren't coming to the review sessions. Someone had a good question. What's the difference between an ion dipole and a hydrogen bond? A hydrogen bond is never going to have to do with an ion. A hydrogen bond is a dipole interaction between either an O or an H, or an O and it's either an O or an N, so an oxygen or a nitrogen with a hydrogen. Sometimes sulfur does it. I wouldn't really worry about sulfur so much in this class, but a hydrogen bond is a dipole bond. So the H is slightly positive and the O or the N is slightly negative, and that creates a hydrogen bond. Hydrogen isn't going to interact with charged molecules, so ions. So it's not an ion dipole. It's a specific type of a dipole. Okay, awesome. So the last question that isn't a problem set question before we get into the exam-like questions was, how can a protein structure change without the primary structure changing? So if we remember correctly, we build structure on top of each other. So we start with primary, and then we go to secondary, tertiary, quaternary. So the way I like to think of it is they're Legos. If you pull out the tertiary level Lego, then it messes up your tertiary, but it also messes up your quaternary because it's on top. But it is possible that you pull that out and you're not affecting primary. And the reason that is because if you say mess up some hydrogen bonding, that might unfold a specific part of the protein. But if we remember, the primary structure is just the amino acid sequence. So even if the protein completely unfolds and it's just the polypeptide chain, that's still the primary structure that we started with, as long as we're not changing an amino acid. The only way our primary structure changes if we break peptide bonds or change amino acids, because the primary structure is literally just the polypeptide chain, the order of the amino acids. So you can mess up all the bonds and unfold the entire protein and still have the same primary structure. That being said, you can also have the same hydrogen bonds between the backbones and have the same alpha helices, beta sheets, but mess up the tertiary structure. But if you mess up your primary structure, you rip out that Lego, you're probably going to mess up all the other secondary, tertiary, quaternary structures. I hope that answers those questions. If you have more questions, feel free to put them in the chat. And that is everything that was in the forum from before. Obviously, ask questions as we go, but we just want to get those out of the way to make sure everyone had their base understanding good. And now we're going to get into some more questions that are questions you might see on an exam or something that has been on a past exam that can help prepare you for the exam. So I'm going to stop sharing this and I'm going to pull up the PowerPoint. Iris, someone just said something in the chat. Do you mind looking at that for me?
Oh yeah, it says last question was mine. Could you elaborate on that a little bit?
Yes, I can. I'm going to put this up real quick. Iris, do you want to take it?
What was the question?
This one was the one about primary structure. And why
Sorry,
primary
can I just
structure?
clarify what I was trying to ask with that?
Yeah, absolutely.
Oh yeah.
So I understand that like you can change higher levels of structure without changing the primary. I was more wondering what would cause the higher levels of structure to change, because when I think of like what causes those structures to form, I think of the R groups interacting, whatever. So I was wondering what would cause like secondary or tertiary structure to change if there weren't different interactions happening between the R groups, like if the primary structure didn't change.
Heat, salt, pH, everything that we talked about in class. I'm so sorry.
Oh, okay, yeah.
So
then,
that
when
can
we were
cause things to
talking
get
about it in
unfolded.
class, when we were talking about PRP, like in that case, I was just kind of confused when we had that discussion because we didn't talk about.
PRP is a very specific situation in which
Okay.
these proteins, when something is misfolded and they come into contact, it misfolds the other proteins so they stack and aggregate. And so the reason we don't necessarily have, it's hard to treat is because it's quite a unique thing. And if you take like a neuroscience class, you'll learn a lot more about it. You don't need to get into exactly why that happens because it's a bit complex, but in that case, it's because these proteins are the same. And when they come in contact, the one misfolded one immediately results in the other one also misfolding, and they start to stack and aggregate. And if that's something that interests you, there's really cool pictures that you can see online of those, what happens to your brain when that happens, because PRP, those are that's Prion's disease, correct? Yeah. Mad-Cowell's disease, same thing. Does that make sense? That one is a little less about the R group interactions, I believe, and just about how the proteins can misfold.
Yeah, that makes sense. Thank you.
Okay. I'm glad thank you for stopping us and clarifying what you were asking. I would hate to waste your time answering a question that you don't have. And so now to the slides. Here is our topic 2, review 2. I thought I was really funny with the slides that I made, so I hope everyone appreciates my memes. And the egg you will see. So our first question, this is the infamous egg question. If you came to my review or my office hours, you will know that I hate this question because I got it wrong all the time in every class ever. So the question is, egg white is primarily comprised of proteins. When egg white is heated to boiling temperatures, it hardens. Cooked egg white can be dissolved by heating it in a solution containing a strong detergent together with a reducing agent. So the question is, what is the molecular explanation for why boiling an egg white causes it to harden? And why can this detergent reducing agent dissolve it? So this is going to be heavily discussion based. So I understand if we're uncomfortable and we want to type in the chat, but I would really love if we could also talk because I think that would really help. And this is like definitely a question that has been on exams in the past. I know this one's really hard and there's a chance that no one knows the answer to it, and that's okay too. But feel free to either write in the chat or discuss. If anyone has any ideas, throw them out there. I don't have just like an answer slide, so this is just kind of a discussion we're gonna have. So, and also here's some cute pictures of some eggs. I'll give everyone a minute to type if they're typing. And if you have no idea, you can put in the chat, I have no idea. I would really appreciate, because I want to make sure that I'm not waiting for people that aren't typing because they don't know. So we have a question that says, do disulfide bonds occur at all between protein aggregates? That is a great question. I'm not sure. While I'm checking the chat, Iris, do you want to Google that? Maybe?
Sure. I wouldn't be surprised, but yeah, I can Google it.
Protein aggregates are very cool because they can definitely happen in a lot of different forms.
Because cysteines would be found, here's my thinking at least, cysteines would be found on the inside of the protein. So when the protein is exposed to heat and unfolds, the cysteines would be exposed, and then they would go find other exposed protein cores and aggregate. So I would think that like any two cysteines of like aggregating proteins would form a disulfide bond.
Yeah, I think you're totally right. I think it can definitely happen. It doesn't necessarily have to happen, but it definitely can if there's two cysteines. I'm not getting any responses in the chat, so I'm assuming that everyone is typing beautiful answers, unless everyone can just send me an I don't know. That'd be really helpful. Okay, thank you. Because if everyone doesn't know, that's totally okay. Oh, there's an answer. So one of these answers, we got a lot of I don't knows, which is why I bring up this question, because this question was the bane of my existence. So I want to make sure everyone understands this. So someone said, does the increase in temperature cause proteins to denature and aggregate together? And then we got another answer that says boiling causes egg white to harden because the heat causes the protein to unfold and then aggregate together due to hydrophobia. I've never heard it said that way. I love that, hydrophobia. I think detergent dissolves it because detergent breaks non-covalent bonds.
I was doing the dishes, so I was in my bra.
You are unmuted, Isabella. I'm going to mute her. Okay, there we go. The heat would break non-covalent bonds, which would cause the protein to unfold and aggregate. All of these are correct. That's awesome. So just to summarize for people that weren't following along there, or people who can't see the chat on the recording, when we boil the egg white, we heat up the proteins. And when we heat up the proteins, it causes our non-covalent bonds, so our bonds involved in our secondary and tertiary structure. In this case, we're focusing mostly on tertiary, to unfold. So our protein's going to unfold. And if we remember, our hydrophobic amino acids are on the inside. The ones that have hydrophobic R groups, non-polar R groups, those are on the inside because they're scared of water. And hydrophilic are on the outside. So when they unfold, we have a bunch of hydrophobic R groups exposed, and they freak out because now they're exposed to the environment, which is mostly water, and they hate it. So they go to find other non-polar things that they can kind of clump with and try to protect themselves. And so that's why they aggregate, because all of these non-polar R groups are finding each other, and that's what causes aggregation, which is why the egg goes from clear to white, because it's clumps of protein, which is kind of weird when you think about it. So next time you eat an egg, think it's clumps of protein. And then the protein, or the detergent question, someone said, I think detergent dissolves it because detergent breaks non-covalent bonds. Yeah, so in the case of the heat, we're breaking non-covalent bonds and unfolding the protein. In the case of the detergent, we have a bunch of aggregates, which are non-covalent bonds. There's like van der Waals interactions going on, and the detergent just dissolves that. So everything's kind of just falling apart at this point. So we aggregated everything together, and now we're dissolving all those interactions. So if we throw in detergent and a reducing agent, the egg kind of just dissolves, the cooked egg white. And we're ignoring the like yolk part right now. We're just talking about the egg white. So if you're bored and want to cook an egg and then throw detergent and a reducing agent on it, you can get rid of it. So yeah, that's really good. And we had a few answers there. So are there any questions on that? What is detergent exactly? I know it breaks non-covalent bonds, but how? You know what? That's a great question. Iris, do you have an answer for that?
Unfortunately, I don't, but I can look it up.
Detergent was always one of those things that Professor Francis goes, we all know what detergent is, right? It just has properties that destabilizes protein interactions. I don't know exactly what it is. Are you raising your hand, Iris? Are you
No,
just
sorry.
chilling? Oh, okay. Detergent, I think that it's just like heat. You like ask what heat is, and like you don't want me to get down to the molecular level, you know? I think laundry detergent, like we call it detergent because it is like chemical detergent because it breaks the bonds and that's what gets the dirt out. I think it's the same thing. So I think detergent is kind of just the word for this thing that breaks non-covalent bonds and destabilizes proteins. I'm sure we could probably get into the nitty-gritty of what it is. But I don't think that's important. Iris, you look
Yeah,
like
so
you're...
what I found was just that detergents disrupt membranes and denature proteins by breaking protein-protein interactions.
They disrupt membranes. So that's the one piece of information we can add to that. So I wouldn't worry too much about the specifics. Just know that what it does. What is the role of the reducing agent again? I understand the detergent part, but I'm still not sure of the role that breaking disulfide bonds plays. Are those two things related? The reducing agent and the...
Well, so disulfide bonds are the strongest bonds you can have in a protein. So reducing agent, if it can break that, it can like completely just unfold the entire protein.
Yeah.
Does that answer your question?
Yeah, so detergent isn't going to break up your covalent bonds. Yeah, so that's a good point. I didn't read the end of the question, obviously. Good job, me. But yeah, we'd be stuck with just like little pieces of the egg white where the disulfide, probably we couldn't even see where the disulfide bonds were. But yeah, the reducing agent is just stronger than detergent. Don't drink reducing agent. Could you just use reducing agent then, or is it specifically for disulfide bonds, not the others? I'm thinking in this case, it's specific to the disulfide bonds. Because obviously we're not breaking the peptide bonds here. So I think that this reducing agent is specific to breaking disulfide bonds. Because at this point we're looking at breaking up tertiary structure and secondary structure, not just breaking all covalent bonds. If you really just want to break all bonds, you just like throw hydrochloric acid on it. That'll pretty much mess everything up. I think the point here is just so that we know detergent's pretty much going to break almost every bond. The disulfide bonds, we just need like a little extra. So I think that this reducing agent in particular breaks disulfide bonds. Okay, if we don't have any more questions, we can move on to the next one. I'm going to give it one more second just to make sure no one has any questions. And of course, we can always come back if a question pops up. Okay. So on to ligands and proteins. Please enjoy my little meme. Everyone can like throw up a laughy face if you think it's funny. I think it's funny. So you are performing experiments with your protein of interest and its ligand. You are attempting to mutate the binding site of your protein to disrupt binding to ligand because you want to observe the consequences of the loss of this interaction. So basically we're trying to mutate where our protein and ligand bind because we want to see what happens if they can't bind. So you make a series of mutations. One in particular gives you surprising results. You observe that the mutation that you made in one of the important interacting amino acids actually increases the protein's affinity for the ligand. Give a molecular explanation for these results. So you mutated a bunch of stuff and suddenly you have a higher affinity. So your things are binding more tightly rather than breaking something and them not binding at all. So can anyone think of any reasons why or what kind of what's happening? What can we do? What can this mutation do that the affinity gets higher, that the affinity increases. I can speak until I'm being recorded and it's going to be on the internet forever, and then I'm just a mess. So if you have no idea, feel free to let me know that. That would be really helpful, but I would love if people could type out some ideas. Oh no. Type out some ideas as to why they think that this might be happening. And this is a great question because it's very similar to your problem set questions that I know a lot of people have questions on that we'll go over later. This also kind of ties into one of your questions, Iris, about if they always have to have strong affinity for each other.
Ah, yes. It all comes together.
Almost like everything we talk about has to do with each other. I don't have any responses yet, so I'm assuming everyone's typing long, beautiful answers. If you're not, just send like an emoji or like an I don't know, so that I know I'm not waiting on 15 people to answer that aren't here. Oh, stop. Someone said KD went down. Why? I want the molecular interaction. I think
Like what could cause KD to decrease?
so. Someone said, I'm sorry, I was reading this whole thing and someone put a smiley face, or no, there's not a smiley face. I read it as a smiley face and I'm just wrong. Someone says if you swap for an amino acid that is smaller, but still has the ability to bind in the same way as the original amino acid, it could make more space for the ligand to sort of nestle in parentheses, or that's what someone said in class. I'm glad you're paying attention in class. And that is one explanation, is that maybe there's more space now and it fits in better and it has the same bond, but it is a little harder to get out of the pocket now. Someone says, could the amino acid be more chemically compatible with a ligand? Like the amino acid is positively charged and the ligand is negatively charged. That is an awesome answer, because if we think, we just talked about the difference between an ion dipole and an ion-ion. So maybe originally it's an ion dipole interaction, and then you mutate it and suddenly it becomes an ion-ion interaction. Those have a higher affinity. That's a stronger bond. So yeah, that's definitely possible. And now my question is, if we made a stronger interaction, we're gonna see who's paying attention. If we make a stronger interaction, then why wouldn't it be like that always? Like if we can mutate it and make it have a higher affinity, then why didn't the cell do that originally? Because cells, they're fighting every day of our lives to be better, to survive better, to be more efficient. So why does it not have the highest binding affinity it can originally? Why can we mutate it to make it stronger? Does someone want to take a stab at that question? Because we answered this question pretty well. There's a few things. Size, we can make it fit better. Higher affinity is not always better. That is exactly right. Why? Bless you, Iris.
Thank you.
Some ligands need to dissociate quickly because proteins are doing a whole bunch of stuff, and it would be inefficient to keep them there after they've completed their function with the protein. That is exactly right. Yeah, ligands are doing lots of stuff. There are some, like we said, transient earlier. Wow, all the questions are coming together. Like we said, there are some transient interactions that need to happen quickly. And then our ligands need to go do something else. We don't want things being together all the time. Say we have an interaction, and when two things come together, they turn on a pathway, and that pathway says it's a cell survival pathway. Let's say that it's cell proliferation. So we turn on this pathway and it tells the cell to grow and to multiply, let's say, or survive. Imagine that is, or yeah, let's do a multiplication pathway, not a proliferation. So a cell division pathway, and this ligand comes together with this protein. Let's say it's a receptor. And then let's say it's really high, like they have really high affinity and they don't really come apart. What problem is that going to cause? Does anyone know? If we have a cell division pathway and the receptor, the ligand and the receptor never dissociate? I know someone knows it. I don't want to spoil my own question. Yes, no, maybe so. No answers yet. Cancer. The answer is cancer. It would be activated forever. Exactly. So if a cell division pathway is activated forever, or at least for a very, very long time when it shouldn't be, suddenly we have cells multiplying when they shouldn't be. And that's one way, there's so many ways, that we could cause cancer, because cancer is uncontrolled growth of cells and multiplication of cells. So yeah, in that case, we want something that's transient. We want the signal to come in and then go. We want to multiply once, then we can't have it be there forever. So that's a great example of why higher affinity is not always better. Some things need to have low affinity, especially like enzymatic stuff and activating pathways and inhibiting pathways we don't want forever, because there's not really anything in our cell that we want to last forever or not. Okay, so I think we're pretty good on this one. Iris, if you have to go, do you have to go?
I do have to go. One note I just wanted to say about the problem sets. So coming from someone that graded your problem sets, just in general, you should be like try to look beyond the question. A lot of the answers were more surface level, and that's okay. It's just week one, and like these problem sets will, get, you'll like understand how to answer them better the more you do, but try to figure out why she's asking those questions, and as much as you can go into like the molecular detail, because that's really like what's important. Yeah, and email me if you have any questions. My office hours are on Tuesdays. That's all on Moodle. All right,
Awesome.
I'm gonna go.
Thank you so much. We have a few more questions, so people that are staying with me. So you don't have to go, but Iris has another, what is it called? Engagement.
Sure.
But yeah, there's a little present for people who watch the entire thing on YouTube, is you have a little snippet of advice. I did not grade problem sets, so I don't know what everyone answered. But we're gonna move on to the next question now. DJKD. I really was proud of this one. And for all my Marvel fans out there that are Marvel nerds like me, we have that. So for antibodies to do their job, and this might be a question you will see because Professor Francis loves antibodies. So for antibodies to do their job, they must bind tightly to their target antigen. If we were discussing the general range of KDs in the cell to be from the nanomolar to the micromolar range, and that's 10 to the negative 9th and 10 to the negative 3rd, where would you expect antibody-antigen interactions to fall in this range, if you know that they have a very tight binding affinity with their target antigens? So this is like an either or question. So would we expect it to be closer to the nanomolar or the micromolar range? Okay, so yes, so we got a lot of responses, and yes, I'm sorry, I don't know why my screen is doing this. Nanomolar. So I'm gonna learn how to use a computer, it's gonna be great. So yeah, so we said nanomolar, so that's the 10 to the negative 9th. And the reasoning is because the higher the affinity, the lower the KD. So 10 to the negative 9th is a smaller number, 10 to the negative 3rd is a bigger number. So since the affinity is higher, the number is going to be smaller. And so we're going to be in the nanomolar range. So that's a pretty easy one, explanation wise. So just understand the relationship between affinity and KD. Okay. Do we have any questions on that one? No. Okay. This might be my last one. Switching it up. So many proteins are regulated through processes that are generally called switch mechanisms, because the proteins are switching between conformational states, and thus their functions are being controlled through this conformational state switching. For each of the 3 different types of protein regulation we covered this week, allostery, post-translational modification, and nucleotide binding and hydrolysis, discuss how each causes a conformational switch. This is literally the questions we just went through, pretty much all combined together. So just throw out one at a time. Some people just pick one at random and maybe write a little blurb about how they cause a conformational switch. It'll probably go quicker if you just write one at a time. You can do all 3. You can just do one. I'm going to give a minute, and I would love to see what you guys come up with. And you can enjoy my picture of this cat being an enzyme and there being an allosteric inhibitor. Oh no, sorry. So someone said allostery changes the active site of the enzyme, and that is absolutely correct. We talked about earlier how, and that diagram on the slides is awesome. If you're not understanding allostery, I would recommend looking at that, copying it into your notes. So allostery, we change the active site of the enzyme by binding somewhere else on the protein. Some sort of protein binds somewhere else, and we change the active site. And like we said, if we change shape, that's a change in conformation. So allostery definitely changes conformation by changing the active site. So that is perfect for that one. Does anyone have anything for post-translational modifications or nucleotide binding and hydrolysis? Anyone want to give me a thumbs up? Do you think next time we do a Thursday review like this, we should have like music between when we're waiting for people to type out answers? Any thumbs up, or anyone think that's a good idea? Maybe we can have some like people submit music on a forum beforehand, and like we pick some songs. I don't know. I don't think anyone responded. Oh yeah, okay, we got a few thumbs up. Awesome. Cool. Maybe we'll do that. That'll be fun. Okay. Post-translational modifications add a chemical functional group, changes overall shape of the protein because something is being added. Yes, absolutely. Thank you for saying that I had a great idea. I appreciate it. And so, yeah, it is reversible. So post-translational modifications are reversible, but the reason that it causes a conformational switch, I mean, you're right. That is why it's called a switch, because it's reversible. You can turn it on and off just like a light switch. But the first one really is hitting the nail on the head, that we're adding something and we're changing the overall shape and chemistry because of it. So if we add a phosphate, that it takes up space, you know, and if it's not charged, suddenly a phosphate group is negatively charged. So yeah, that's exactly right. That if we add it, we're changing our shape, we're changing our chemistry. Does anyone want to take a stab at nucleotide binding and hydrolysis? Is anyone typing it out? Or we went over it, so maybe this is a good time to test yourself if you understand. I'll give it another minute. So yeah, I got at least 3 thumbs ups on the music thing. So I'll talk to the other TAs and try to get that to happen. I'm gonna give it like another 30 seconds, see if anyone has any ideas on this one. I say 30 seconds as if I know what 30 seconds is off the top of my head. Okay, no one oh, yes. So the shape of the protein would be different when it's binded to or bound to GDP. So this is true. So yeah, you're thinking of the example, the GTP, GDP example, and that's absolutely right. I'm going to go a little more in depth. Isn't this one related to hydrolysis, things being taken off? GTP, GDP. Yes. So those are both in the right realm. So GTP is guanosine triphosphate. So that is a guanosine, which is one of our nucleotides. Remember, we have 4, our 4 letters. Well, 5 because uracil, but your ATCG. So G is guanosine, triphosphate. So it's a guanosine with 3 phosphates on it. So that's where the nucleotide part comes in. So binding and hydrolysis. So when GTP is bound to a protein, in this case, GTP is going to activate it. So it's bound, and when it's bound, it changes the conformation in a way that activates a protein, right? And then we hydrolyze, so we remove the phosphate and we make it guanosine diphosphate, which is 2 phosphates. And when we do that, the structure changes, which means the whole conformation and shape changes. And when it's in that state, then it's not active. So that is how it's a conformational change, is that GTP versus GDP, you take off that phosphate, it's literally a different shape. And so that'll change conformation. And what we looked at earlier is that GTP activated and GDP was inactive. So it's a little different than adding a phosphate to activate or deactivate. We're using a whole nucleotide, and GTP and ATP provide energy. So it's a little different. Do we have any questions on that? I know that's a lot easier to understand with a picture. And if you want to go over that again, we definitely, the beginning of this recording, we went over that for sure. So I would highly recommend rewatching that if you still have questions. So that's that one. And so do we have any questions? And this is my favorite new meme. It says I used to have all the answers. Now I don't even understand the questions. And I feel like everyone in this class can relate to that at least once. I definitely did. So before I stop the recording and we get into problem set questions, do we have any questions on material that was unclear that we went through today? The Kahoot, which I know was not recorded, or any of the questions we answered on the forum, or any of these questions that we had up. Like I said, these are the types of questions you're going to see on the exam. So these are going to be the kind of things that you're going to want to be able to answer. And we haven't provided this type of review in years past, so we're trying it out to try to give people more preparation so that they feel like they're ready for what they'll see on the exam. So how are post-translational modifications added to the protein? Other proteins. The answer is there's sites for it. So a kinase adds a phosphate. So a kinase is an enzyme, and you'll talk a lot about kinases. A kinase is an enzyme that adds a phosphate group to a protein, and it adds at a very specific spot. So there's like serine threonine kinases that add on serines or threonines. I'm actually looking at one of those right now in my cancer genetic class to treat cancer. So they're very specific. And so a kinase can add a phosphate, a phosphatase removes a phosphate. There's similar stuff for ubiquitin and acetylation, but kinases and phosphatases are probably what you're going to talk most about. So yes, to add those modifications to a protein, we use another protein, and that's an enzyme. Does that answer that question? And like I said, they're added to very specific places at very specific times. So, but we haven't gotten into that necessarily yet. You'll definitely get into RTKs, different kinds of kinases and how they function and what they do. Okay, that's an awesome question. Are there any other questions? No one typed anything, so I'm going to wrap this up. So here's my thank you. Our emails are at the bottom. I will be sending out these slides as soon as the recording is uploaded on YouTube. I'm going to send that out with the link. Thank you all so much for coming, watching this recording. And if you want to stay put and ask questions you weren't comfortable asking on the recording or go through problem set questions, that's what we're going to do now. And for those of you watching the recording that wish you were here to ask problem set questions, Tuesdays and Thursdays at 7 p.m. Feel free to come. That is not what I meant to do.